Hello and welcome back to my channel. My name is Shirsha, and in this particular lecture series we are going to be talking about protein purification. So because protein purification is such a vast topic, we will not be covering it all in one lecture, but rather I would be uploading multiple lectures to talk about the different aspects of protein purification, what are the various commonly used techniques, and so on and so forth. So without further ado, let's dive into the topic. So to begin with, why do we need to purify a protein? Why at all do we require a purified protein? Protein purification is a series of processes intended to isolate one or a few proteins from a complex mixture, usually cells, tissues, or whole organisms. So say for instance, there is a particular protein which is of great interest to you and you want to study that protein. Now it would be much easier to study it if you are able to isolate that protein from the entire cellular system, because the cellular system comprises of hundreds and thousands of different proteins, and it would be quite difficult to specifically study one particular protein in it, especially depending on the kind of studies you want to do. Say for instance, you have a new protein and you want to know the sequence of that particular protein. So how do you do that? You want to know the amino acid sequence. So in order to do that, you first have to isolate the protein. So in this scenario, let us assume that you don't really know the gene encoding for a protein. You know that there is a certain protein, say let's call it A, that is interacting with another protein B, and you want to know the sequence of that particular protein A. You want to identify the particular protein A. So if you have already gone through my previous lectures on co-immunoprecipitation, so co-immunoprecipitation is nothing but a small scale protein purification technique whereby by doing co-IP, you can identify this protein A which is interacting with your known protein B. And once you have this protein A at hand, you can then do further analysis to identify the primary amino acid sequence of the protein. And from that, you can then simply determine the genetic sequence that would be responsible for encoding for that particular protein. So, protein purification is vital for the specification of function, structure, and interactions of the protein of interest. So, what do I mean by specification of structure? By you can solve the structure of a protein by different techniques like cryo-electron microscopy or X-ray crystallography. But in order to do so, you require purified protein in hand to begin with. So protein purification can help with structural determination of a protein. What about interactions of the protein? So if you have the purified protein at hand and if you can modify it by adding to it some tag, so that you can then perform co-immunoprecipitation with the purified protein, then you can simply perform a co-IP experiment from cell lysate where you're adding your purified protein with the tag and you're adding a cell lysate, and you can identify what other partners are interacting with your protein of interest. You can also study the function of that protein. You can study the physicochemical characteristics of that particular protein, like how stable a particular protein is, at what temperature it is melting, so on and so forth. So the purification process separates the protein and the non-protein parts of the mixture. First, to begin with, so because a cell comprises of not just proteins but other biomolecules as well. So the first step would be to separate the protein from the non-protein parts, and then finally to separate the desired protein from all of the other proteins. The separation steps employed usually exploit differences in the protein size, the physicochemical properties, binding affinity, and biological activity. Now, as we discuss more and more about this topic, it will become clearer how these different properties are exploited to purify a protein. So this is a structure of interleukin 12 bound to its receptor. So this is a very classical example of one of the applications of protein purification, which is to determine the structure of the protein. And in any day, structural determination is the final step of completely understanding a protein. because once you know the structure, you know how the various amino acids and how the various atoms within the amino acid are physically arranged and what spatial location they occupy. Now, one of the primary ideas that should be cleared before we delve in further details into protein purification is the difference between analytical versus preparative purification. What do each of these terms actually mean? So, protein purification can be either preparative or analytical. So, what is preparative grade purification? Preparative purifications aim to produce a relatively large quantity of purified proteins for subsequent use. Examples include the preparation of commercial products like enzymes, for instance lactase, nutritional proteins, and certain biopharmaceuticals like insulin. So yes, protein purification has applications in the production of biopharmaceutical compounds as well, for a very common example of which is insulin. Several preparative purification steps are often deployed to remove any byproducts which are essentially unwanted protein contaminants or non-protein contaminants such as host cell proteins, which poses as a potential threat to the patient's health. So when you're thinking in terms of purifying a protein for biopharmaceutical applications, you require very high grade pure protein, because otherwise these unwanted contaminants will have unwanted side effects in the patient once you subject them to the pharmaceutical product. Next, we have the analytical scale of protein purification. Analytical purification produces a relatively small amount of protein for a variety of research or analytical purposes. So essentially, the purification that we do in laboratories can be classified as analytical purification, where we purify a protein so that we can do further studies with it, and we do not require such huge or large quantities of protein as we would otherwise be purifying in preparative grade. So these purification or analytical purification includes identification, quantification, and studies of the protein's structure, post-translational modifications, and functions. So pepsin and urease were the first proteins to be purified to the point that they could be crystallized. Do keep in mind that if you want to go for structural determination of a protein, then you require very pure protein, and pepsin and urease were the first ones which could be purified to that level of purity that they could be crystallized. So the first step of protein purification is cell lysis, and in this particular lecture we are going to be talking in detail about the different techniques of cell lysis, when to select which technique, and what are the advantages and disadvantages of each of these techniques. Now, why do you need to lyse a cell? Obviously, because your protein would be expressed by these living cells and you will be having the protein in the cytoplasm of the cell. So in order to be able to purify the protein, you first have to lyse the cells. So this cell lysis falls under preliminary steps of protein purification, and we could also call it as extraction. So if the protein of interest is not secreted by the organism into the surrounding solution, the first step of each purification process is the disruption of the cells containing the protein. Do keep in mind that not all proteins that are purified from the cytoplasm of the cell or nucleus of the cell if it's a nuclear protein. Some proteins are also secreted by the cells depending on if they have a signal sequence that will promote secretion, or depending on whether they require to be secreted in order to allow for proper folding and proper structure of the protein. Under such situations, we will be purifying the protein from the media into which the protein has been secreted. Say for instance, you require certain disulfide bridge formation in your protein, and we know that in order for this disulfide bridge formation, you actually require an oxidizing environment. The environment within the cell in the cytoplasm is reducing in nature, and over there as a result disulfide bridges would not form. So if your protein requires disulfide bridge formation for proper folding, and in order to take the final proper fold, you would then have to ensure that your protein is secreted by the cell. So in order to ensure that, you would typically add some signal sequence tag at the beginning of the protein, and you would then be purifying the protein from the medium into which it was secreted. But that is just a small fraction of the cases. In majority of the cases, your protein will be intracellular, and you have to disrupt the cell to release the protein. Now, depending on how fragile the protein is and how stable the cells are, one of the following methods can be used: repeated freezing and thawing, sonication, homogenization by high pressure, for example by using a French press, homogenization by grinding, for example by using a Downs homogenizer, permeabilization by detergents, for example Triton X100 and slash or enzymes like lysozyme. So the cell debris can then be removed by centrifugation. So once you have lysed the cells and you have released the intracellular contents, you have to remove the cell debris so that they do not interfere with the subsequent steps of protein purification. So the cell debris can be removed by centrifugation because by virtue of being heavier, they would settle down and your soluble fraction will remain in the supernatant and your protein will be in the supernatant. Proteases are released during cell lysis, which will start digesting the proteins in the solution. So this is a very key and important factor to keep in mind. If the protein of interest is sensitive to proteolysis, then it is recommended to proceed quickly and to maintain a low temperature. You keep the extract cool and you keep it on ice during all of the steps. And if you have to perform any step which cannot be performed on ice, then you have to perform the step in a cold room chamber where the temperature is maintained at 4 degrees centigrade. So why is this done? This is done to slow down the digestion that would be otherwise ensured by these released proteases. Alternatively, one or more protease inhibitors can also be added to the lysis buffer immediately before cell disruption. So this is actually commonly done in laboratories, whereby when you're going to or when you're preparing the cells for lysis, before you subject the cells to any of these processes of either sonication or downsing or adding of detergents or enzymes, you will always add certain protease inhibitors to the cell lysis buffer. Commonly used protease inhibitors include PMSF and benzamidine, but these are just two examples, and there are several different kinds of protease inhibitors that are available commercially. Sometimes it is also necessary to add DNAs in order to reduce the viscosity of the cell lysate caused by a high DNA content. Mostly in case of large scale protein purification, DNA's addition is typically not required. But say for instance, when you want to isolate the um, protein from any mammalian cell culture, for example, so the DNA or the genomic DNA content that will be released upon cell lysis will increase the viscosity of your solution several fold. So to reduce that, DNase is required to be added. So now taking a look at each of these processes, to begin with, repeated freezing and thawing. Now repeated freezing and thawing is one of the simplest yet effective processes of cell lysis. The freeze-thaw process represents a pretty or a very mild homogenization procedure. Why is it mild? Because you're not really subjecting your cells to any physical pressure or to any physical force, or you're not adding any enzymes or detergents. It is commonly used to lyse bacterial and mammalian cells. The technique involves freezing a cell suspension in either a dry ice or ethanol bath or freezer, and then thawing the material at room temperature of 37 degrees Celsius. This method of lysis causes the cells to swell and ultimately break as ice crystals form during the freezing process and then contract during thawing. So this is very similar to what we observe when rocks are broken down as a result of this repeated freezing and thawing. So when you first freeze the cells, because you have freezing the cells very quickly in either dry ice or in an ethanol bath, so this quick freezing promotes the formation of ice crystals. Now once you have these ice crystals, so as you thought, these ice crystals are taking up space. They will result in an increase in volume. Now once you thaw them and these ice crystals melt to give rise to water, this will actually empty up the space that was taken up by them. And this repeated process of freezing and thawing will lead to the lysis of the cells. Because at first the cells will swell up, and once you thaw them, there will be a space, an empty space created within the cell. So now because the cell would be in the lysis buffer, this will promote the inward movement of this buffer from the outside of the cell to the inside of the cell, and this eventually will lead to the swelling and bursting of the cells. Multiple cycles are necessary for efficient lysis, and the process can be quite lengthy. However, freeze-thaw has been shown to effectively release recombinant proteins located in the cytoplasm of bacteria and is recommended for the lysis of mammalian cells in certain protocols. So essentially, you have your cells submerged in a buffer solution, you freeze them, so you have dry ice or on either dry ice, or you can freeze them with low temperature, say by directly putting them in a -80-degree freezer, and then you thaw at room temperature. And you repeat this process several times to ensure effective lysis of your cells. Although it has proven to be effective for several cases, it is not very commonly used, and other methods of cell lysis are more preferred because of their higher efficiency as compared to repeated freezing and thawing. However, if you are purifying some sensitive proteins which would otherwise be at the risk of being denatured by the other processes of cell lysis, this process can be then utilized because of its mild nature. So the advantage of this process is that it is an inexpensive technique, but the major disadvantage is that it is very time consuming because you have to subject the cells to repeated rounds of freezing and thawing, and it cannot be used for extracting cellular components that are sensitive to temperature. You do have to keep in mind that if your protein is sensitive to this repeated freezing and thawing, which most proteins are, then this particular procedure has to be avoided, because this repeated freezing thawing cycle will be affecting your proteins as well, and that is not a good outcome, or that is undesirable, I could say. So the next type of cell lysis method, which is the most commonly used for purifying proteins from bacterial cells, is sonication. Do keep in mind, sonication is not commonly used when purifying proteins from either mammalian cells or insect cell cultures. So sonication is the act of applying sound energy to agitate the particles in a sample for various purposes, say for instance like the extraction of multiple compounds from plants, microalgae or seaweeds, or in our case for purification of the protein from bacterial cells. Ultrasonic frequencies of more than 20 kilohertz are usually used, leading to the process also being known as ultrasonication, or ultrasonication with a hyphen in the middle. In the laboratory, it is usually applied using an ultrasonic bath or an ultrasonic probe, which is commonly referred to as the sonicator. So this is what a typical sonicator looks like. You have this machine using which you can set the frequency at which you want the ultrasonic waves to be released. You can set the on and off time, that is how long you want the wave to be released and then how long you want it to be off. Typically, because sonication generates a lot of heat, so your sample which is being sonicated has to be kept on ice. It is an absolute must. Otherwise, the heat released from sonication can denature your proteins of interest. Also, because of this heating issue, sonicators typically operate in an on and off cycle, where you typically say for instance if you are keeping the sonicator on for 15 seconds, it will be off for twice the time, which is 30 seconds. So if you keep it on for 10 seconds, then it will be off for 20 seconds. So you have this on and off cycle which goes on repeatedly, and all of this can be set using this machine which is attached to the sonicator probe. So this is your sonicator probe which is immersed in the solution, or rather in the cells suspended in the lysis buffer. Also keep in mind that this probe, it's preferred that it is immersed 3/4 of the way of the cells resuspended in the lysis buffer to ensure most effective and most efficient sonication. But do be careful that this sonicator probe does not touch the bottom of your glass beaker, because then otherwise your beaker would simply crack. Yes, I forgot to mention, all of these sonication procedures are carried out in glasswares and not in plasticwares. So basically what happens is that if we take a look at the mechanism of how ultrasound waves induce cell damage, so you have these ultrasound waves which are coming and hitting your cells, and this initiates the formation of pores in the cell. Now as pores form in them, the water molecules that were present outside in the cell lysis buffer will enter within the cell, and this will cause the cells to swell up, and finally there will be rupture of the membrane and release of the cellular contents. So this is quite similar to what was happening in repeated freezing and thawing, whereby this influx of water molecules into the cell was induced by the formation and subsequent thawing of ice crystals, and here it is induced by these ultrasound waves which are inducing pore formation in the cells or in the cell membrane. So coming to the various advantages and disadvantages of the sonication procedure. So the advantage is that it is independent of cell type. You can use it not only for bacterial cells but also for mammalian and insect cells, but preferably it is not used for mammalian and insect cells because you can lyse them by other methods. But well, I would again like to add over here that it would depend on what kind of protein you are aiming to purify. If you want to purify certain cytosolic proteins, then you can always go for sonication with insects as well as with mammalian cells. But other milder methods of homogenization are also available that could be applied to them. But because plant cells, for example, or bacterial cells, they have a thick cell wall, so sonication helps with efficient disruption of the cell wall. It has high lysis efficiency. It shears the chromosomes as well, eliminating the need for nucleus treatment. Because when you are sonicating, the genomic content would also get sheared. So the viscosity that we were talking about in the initial slides, that is generated due to the genomic content released upon cellular lysis, that issue can be circumvented by sonication. Disadvantages: it produces a lot of heat which needs to be mitigated to prevent damage to sensitive proteins, and hence you always sonicate while keeping a sample on ice, and you have this alternating on and off cycles. It produces cellular debris, but that is a very minor disadvantage. You can always remove the debris by centrifugation, and it's a loud process and requires sound mitigation. So you have to take care of the noise that is generated during the sonication procedure. This noise is not really good for human ears, so always take adequate precautions. So the next method that we are going to discuss is homogenization by application of high pressure. High pressure homogenization uses force such as turbulence and cavitation along with high pressure to create a consistent and uniform sample. So what do I mean by that? We will clarify. It is an efficient means of cellular disruption whereby a sample is subjected to ultra high pressure as it is forced through a narrow orifice. As the sample exits the orifice, stress forces like cavitation and shear forces are generated, and these forces then ultimately lyse the cell. Because of its powerful pressure, high pressure homogenization is well matched with organisms like bacteria, yeast and fungus, where you have a tough cell wall that needs to be lysed. And such high pressure homogenization is typically not required for systems that do not have a cell wall, like for mammalian cells or for insect cells. A commonly used high pressure homogenizer is the French press. So this is what a typical French press looks like. And if we are to take a look at the working principle of the French press, so essentially this green thing is your sample, basically your cells suspended in the lysis buffer. You put it within the French press and you have this plunger with which you press down. Now as you're pressing down with the plunger, there is this very narrow and very thin orifice present at the bottom through which your sample will exit. Now, as it is exiting through this narrow orifice, it will create a lot of pressure on the sample, because over here you're applying force on it and you have this very narrow orifice through which it is exiting. So based on the principles of physics, it will generate a lot of pressure on your sample, and along with the pressure there will be the generation of forces like cavitation forces and shear forces, which will ultimately lead to the lysis of the cells. So the advantages of this method includes high efficiency and the fact that it is not cell type dependent. It can be applied to any system that you want to purify your proteins from. The disadvantages is that it generates a lot of heat, but the amount of heat generated is lesser or comparable to the amount of heat generated with sonication, and this heat could damage your intracellular products. It is an expensive method because you need the French press equipment and you can't just do it without it. And it is difficult to purify the lice samples. Well, it is not always difficult. Sometimes it might be difficult, but not really. This particular disadvantage, we can put it aside and it can be easily circumvented. So the next method is homogenization by grinding. So this method disrupts cells through the use of the force of friction when the sample is placed between two hard surfaces. The sliding action of one or both the surfaces then causes a mechanical disruption. Although it may be employed for frozen, wet or dry samples, grinding is most effective with solid samples. The dual forces of pressure and shearing force cause a ripping and tearing that effectively disrupts the tough cell walls. While a mortar and pestle is the best known grinding tool, other options like glass homogenizers and grain mills are also used. So mortar and pestle is a much more tougher method, I could say, because it would generate greater amounts of pressure and shearing force, and therefore a mortar and pestle is preferred for disruption of plant cells where you have the presence of a thick cell wall, because you can apply more pressure and more force when using a mortar and pestle. But when using something like a Downs homogenizer, this is much milder when compared to a mortar and pestle, and the pressure applied over here is lesser and more regulated compared to a mortar and pestle, and therefore it can be used for more sensitive samples whereby you don't have to deal with a thick cell wall like insect or mammalian cells. So coming to the advantages and disadvantages. So the advantages of homogenization by grinding include the fact that it works well with tough tissue samples like plant tissue samples, whereby you can use a mortar and pestle, as well as with soft samples like where you can use a Downs homogenizer. So it can be used with both kinds of samples, both tough and soft samples, where you have different kinds of grinding techniques or grinding equipments available to help regulate the amount of pressure or amount of force that you're applying on your sample. It is compatible with small volumes and you can reconstitute in a buffer of your own choice. What do I mean by that? That you can basically simply, after you grind it, say for instance you have your plant sample, say for instance leaves, and you grind it in the mortar pestle. Once you have ground it, you can simply resuspend the sample in your buffer of your own choice. Disadvantages is that it is time consuming and reproducibility may vary because it is dependent on how you're grinding it. It is completely dependent on the user who is performing it, because you're not really using any machine for it. So therefore the reproducibility may vary. Next we have permeabilization by detergents. Now, detergent-based lysis results from the incorporation of detergent into the cell membrane, which will ultimately lead to the solubilization of the lipids and the proteins in the membrane, creating pores within the membrane and eventually leading to full cell lysis. So basically, as we know, detergent molecules have a hydrophobic head and a hydrophilic tail. And if you think of your cell membrane, it's a phospholipid bilayer with a hydrophilic outside and a hydrophobic center. So these detergent molecules, by virtue of their amphiphilic nature, will insert into the membrane, where the hydrophobic tail of the detergent molecule will insert into the hydrophobic center of the membrane, and this will therefore help in creating pores in the cell wall. In the cell membrane. So many different detergents are used for this purpose, including ionic, non-ionic and zwitterionic moieties. Now what kind of detergent you will be using is dependent on what cellular system you're going to purify your protein from. If you are purifying it, and also it will depend on what kind of protein you're purifying. For instance, if you are purifying any cytosolic protein and your main aim is to simply disrupt the cell membrane completely, then you can use harsher detergents like ionic detergents. However, if you want to purify any membrane protein where the protein is embedded in the membrane and you want to isolate the membrane, then you will be using milder non-ionic detergents. So yes, like I discussed previously, so, like I was already saying, once you add the detergent molecules, so these detergent molecules will insert themselves in the phospholipid bilayer, as you can see over here, and then they would break apart the phospholipid bilayer, where you will have just the detergent micelles. You will have the detergent micelles containing the phospholipid from the membrane, and you will have the detergent micelle containing the membrane proteins. So by this particular method, or by using detergents, you can actually purify membrane proteins from the cell. So these are some of the detergents that are commonly used for protein purification. You have SDS, or sodium dodecyl sulfate, which is an anionic detergent. It is a very strong lysis agent. It works with majority of cell types. However, it is not suitable for sensitive protein extraction because of its denaturing properties. Then you have the Triton X's, the Triton X 100 and 114, which are non-ionic detergents, and because they are non-ionic, like I previously mentioned, they are non-denaturing and they are mild lysis reagents, and they work well when performing protein analysis. Next, we have NP40, which is another non-ionic detergent. It is also non-denaturing in nature and is a mild lysis agent. It is good for isolating cytoplasmic proteins but not nuclear proteins. Then we have the tweens, tween 20 and tween 80, which are the most commonly used ones. These are also non-ionic detergents, which are mild lysis agents, good for cell lysis and protein isolation. And finally, we have the chaps and chapso, which are zwitterionic detergents, also mild lysis agents, good for protein isolation. So what particular detergent you will be using for your cell lysis will depend on the system. It will depend on the protein that you're purifying, and it will obviously require some optimization before you can finally zoom in on a specific detergent and a specific amount of detergent that needs to be added. However, do keep in mind that if your aim is to purify membrane proteins, then neither of these detergents can be used for that particular purpose, and very specific proteins are available. Very specific detergents are available for the purification of membrane proteins in micelles. So coming to the advantages and disadvantages, the major advantage is that it's suitable for protein release, but the disadvantage is that it is not suitable for isolating sensitive enzymes and proteins, since reagents used are very expensive. So if you want to purify a large amount of protein, in that case detergent or using detergents is not a very suitable or favorable choice, because these detergents are typically very expensive. Now, it is difficult to remove the chemical reagent from the sample after lysis, and lower efficiency, as complete lysis is not possible with using only detergents. So typically what is done is that a combination is used. Like for instance, you can keep detergent in the lysis buffer and then you can sonicate the cells. Or you can keep detergent in the lysis buffer and then you can go for down homogenization. So this combination of different cell lysis techniques helps achieve efficient cell lysis, and it has to be optimized not only for your system but within the system for different proteins that you're purifying. The final method that we are going to talk about for cellular disruption is by using enzymes. Enzymatic lysis is a biological cell lysis method in which enzymes like lysozyme, lysostaphin, zymolase, cellulose, protease, or glycanase are used. So typically you will be using enzymes for cell lysis when your target cell has a cell wall, and these enzymes aim at disrupting the cell wall. Most of these enzymes are available commercially and can be used for large-scale lysis. One advantage of enzymatic lysis is its very high specificity. Now, because these enzymes will be specifically targeting the cell wall of a specific kind or of a specific chemical nature, this ensures high specificity. This method is commonly used to lyse bacterial or plant cells where a cell membrane, this is a typographical error, where a cell wall is present. For example, lysozymes are used for bacterial cell lysis, whereas chitinase can be used for yeast cell lysis. Because why? Lysozymes, what do? They attack the peptidoglycans, and bacterial cell wall is made up of peptidoglycan. Chitinase can be used for yeast cell lysis. Why? Because the yeast cell wall is made up of chitin, and so you're going to use chitinase for it. Pectinases can be used for plant cell lysis. Why? Because the plant cell wall contains pectins. So you will be using specific enzymes that target the specific compounds that make up the cell walls of different cell types. So this is a generic example of bacterial cell wall digestion by lysozyme. So the bacterial cell wall is made up of peptidoglycans, which comprise of repeating units of NAG and NAM, and the lysozyme comes and cleaves between these NAG and NAM residues, so or these NAG and NAM repeating units. So the advantage of permeabilization by enzymes is that it can be very specific for cell types and it is suitable for extracting proteins. The disadvantages include that complete lysis is not possible, expensive reagents because obviously you're using enzymes, so they are very expensive to obtain, and it has to be used in combination of detergents for bacteria. Now if you want to use enzymes for bacteria, then because their cell wall is pretty rigid, so just enzymes will not do it. You have to use it in combination with detergents. So like I already said previously, the ideal method for cellular disruption has to be determined by trial and error. It is not same for all systems, and even within a system, say for instance you purify your proteins only from bacteria, but the same method of cellular lysis cannot be applied to all proteins that you're purifying from bacteria. Different proteins require different methods of cell lysis, and this will depend on the sensitivity of the protein. It depends on the protein that you're purifying, where it is localizing within the cell will also play an effect in determining which method is more suited. Also, say for instance within bacteria, if you're using different strains, then different strains might sometimes require different methods of cellular lysis. So in order to obtain efficient cell lysis, the first step would be to always optimize the conditions that are best suited for a particular system and within the system for a particular protein. Most commonly from experience I can say that generally not any one of these methods is used for cell lysis, but rather a combination of different methods are used. Like detergents and enzymes are commonly used, detergents more commonly than enzymes, and these are always used in combination with one of the previous methods that we discussed, like either homogenization by French press or homogenization by grinding or for sonication. The repeated freeze-thaw cycle method of cell lysis is less commonly used, but it is used for purification of proteins which are very sensitive to harsh conditions. So that is all that we had to discuss about cell lysis. In the coming up lectures, we will be discussing about how to separate the soluble fraction which contains your protein of interest from the cell debris. which is essentially by centrifugation, and we will be discussing in detail about the different types of centrifugation, when to select which kind of centrifugation. We would also be talking about ammonium sulfate precipitation, which is a method commonly used to remove unwanted proteins from your soluble fraction and to leave out only your protein of interest. So it's basically a procedure for cleaning up the soluble fraction so that you have just your protein of interest with maybe few other contaminating proteins, and a majority of them have been removed by this precipitation method. So this is all we had to discuss about cell lysis. I hope that I was able to convey my point, and in order to complete the lecture series, do stay tuned, and I will be shortly uploading the videos on the subsequent steps of protein purification. In the meantime, if you have any questions or any doubts, please feel free to leave it in the comment section and I will definitely get back to you as quickly as I can. If you like this video, then do like it and subscribe for more such videos and share it as well. So until next time, bye.